Hi and welcome everyone. This is Haitham Taha. I will present my project, The Drivers of Brucellosis Transmission in Jordan, a system dynamics approach. In the beginning, I will provide a brief background about the research topic. Brucellosis is a serious public health and animal problem distributed worldwide. It's highly prevalent in the Middle East region. Annually, more than 500,000 cases of brucellosis are reported from the endemic regions. Jordan has a high brucellosis burden in both sheep and human. Brucellosis prevalence was 22% in sheep flocks. Vaccination is the only formal government response to control sheep brucellosis. However, it is not effective. Only 1.5% of small ruminants were vaccinated. Despite vaccination, why is brucellosis a persistent public and animal health issue in Jordan? What are potential more efficient strategies to control it? In order to answer these questions, we have to understand the dynamics of drivers of brucellosis transmission in human and sheep. To answer these research questions, systems thinking approach were applied to use system dynamics modeling methodology to develop causal loop diagram and stock and flow model. Finally, to suggest policy and scenario designs. To understand the system dynamics, we identify the current problems by reviewing data from the available literature and governmental reports. We identified relevant stakeholders to provide valuable data inputs and enrich the modeling process by their knowledge and understanding. To develop the causal loop diagram, we conducted interviews to capture stakeholders' embedded mental models and system structures. The overall causal loop diagram modeling process involved 5 interconnected stages. The modeling starts with the thematic analysis of interviews data to create the first causal loop diagram. The first causal loop diagram was refined, enhanced, and then transformed into a structured causal loop diagram by constant checks with the stakeholders. The structured CLD will be used as a reference for the next stage, stock and flow modeling. Next, we developed the stock and flow structure, tested the model, ran sensitivity analysis, and finally proposed policy and scenario designs. We used Stella Architect as a modeling software. We created a subsystem diagram that incorporates 5 sections involved in brucellosis transmission. These sectors are found dynamically interacting in the system. The previous diagram is the foundation to develop this causal loop diagram or dynamic hypothesis. This causal loop diagram involves 2 reinforcing feedback loops and 6 balancing feedback loops. Reinforcing feedback loops are first, brucellosis transmission among sheep, labeled R1 in red, and second, brucellosis transmission between sheep farms and sheep markets, labeled R2 in violet. Balancing feedback loops are related mainly to brucellosis control, people susceptibility, and veterinary services. Based on the previous causal loop diagram, the following structures represent the stock and flow model that represent the brucellosis transmission system. The first is the human infection structure, mainly involve farmers. The structure is a modified population model, involves susceptible, infected, and recovered population. We added aware population. Susceptible people can be infected through contact or consumption. Awareness can protect people, therefore they are not susceptible anymore. Finally, infected people are recovered. The second part of brucellosis model is an age and gender structured sheep population model. This involves 3 different structures: ewe, ram, and gestation. The first is the ewe infection model structure. This model allows births, aging, and deaths. sheep can be vaccinated for a period of time. Importantly, the model accounts for infections. Finally, the model allows buying sheep from markets and illegal transboundary entry into farms. The second is the ram infection model structure. This structure is similar to U structure, except rams cannot be vaccinated. The third part is the gestation and lambing structure. This model counts sheep in gestation and lactation periods. Importantly, it models births and abortions per each season. Additionally, we counted ram or U numbers get infected in each infectious mating in each season in the following infection probability structure. In the food safety section, we modeled food safety and counted contaminated dairy products consumed by farmers. Contaminated dairy products are an important input in the human infection model. In the public health section, patients with symptoms of brucellosis are an important part of the public health and brucellosis reporting system. Similarly, in the veterinary section, farmers' visit to the veterinary clinics represent a cornerstone step to identify and confirm sheep brucellosis and to provide services like sheep vaccination. The stock and flow model produced in this stage reflects on the causal loop diagram developed earlier. The sectors involved could be in dynamic interactions and have embedded relationships. Therefore, the model structure could explain the dynamic behavior of brucellosis transmission. In this comparison, the simulated model behavior is closely analogous to the observed system behavior. This could support the dynamic hypothesis we assumed between the involved sectors. Several leverage points were identified through the sensitivity analysis. The system was sensitive to changes in the following parameters: slaughter, people's awareness, farmers' visit, test and cull, market restrictions, and vaccinations. In collaboration with the stakeholders, the following 8 scenarios were designed to control brucellosis transmission. The effect of each scenario on brucellosis transmission was assessed on a radar graph. The scenarios were compared and ranked according to the overall reduction the scenario produces on human and animal brucellosis cases. Finally, in conclusion, we developed a tool for policy design enable stakeholders to analyze scenarios. Sheep vaccination was not utilized efficiently and by itself is insufficient to control the disease. Farmers' visit to veterinary services are critical in sheep vaccination rates. Test and cull infected sheep is the most efficient brucellosis control strategy, however, should be considered only after a thorough assessment. A greater control might be produced by utilizing multi-sectoral relationships embedded in the system. Thank you very much.